Okay, good morning, class. Welcome to this new course unit uh, of Introduction to Pharmacology. First, our learning objectives. You should be able to define pharmacology, outline the scope of pharmacology, explain the branches of pharmacology, describe the naming of the drugs, and describe the processes of drug development. Let's look at the introduction to pharmacology. We said pharmacology is the study of drugs and their interactions with living systems. That's to say, the systems within our bodies. The scope of these studies, we have studies physical, chemical, and properties of drug. That is the chemistry. So we shall be studying the physical and chemical properties of the drug. We shall also be studying the effects of drugs on the body's organ systems, that is the physiology and anatomy, how the drugs interact with the physiological system and also anatomical setup of the body systems. Studies molecular composition of a drug and its effect on a drug structure, that is biochemistry, and then also study the effects of drug on human behavior. So there is a psychological aspect as well. Uh, let's first look at the sources of drugs. Where do we get drugs from? Basically, most of the drugs that we use, some of them are obtained from natural sources, like the plants, for example. Example, digitalis, the quinine itself. Some of you already familiar. We are using quite a lot of artemisinin combination medications currently for as antimalarials. You can as well try to check how the artemisinin plants looks like. They are actually one of the major uh, uh, a substance that people produce as a way of livelihood just because the pharmaceutical industry is making quite a good use of it from our farmers. So this is something that is really one of the best things to grow in these days. For the animals, we know that some of these drugs are derivatives from the animal. The example is the insulin we use. Okay. We also know the antivenom, the snake antivenom. Most of these are manufactured. They are extracted from animals. Uh, for example, the snake venom is amplified. We make more copies of this in other animals, like in the horse. So it is like getting the substance and then it's injected into another animal to produce more antis for the snake venom. Synthetic sources, we say chemical manipulation and engineering. For example, our penicillins that are commonly used from the narrow spectrum penicillin to the extended spectrum penicillin, the amoxicillins and the ampicillins that we use currently. All these are groups of the penicillins. Also, aspirin is one of the synthetic drugs. And then we have the biotechnological sources. These ones are made through the process of genetic engineering and cell cultures. We also have some insulin analogs are made through the process of genetic engineering and the monoclonal antibodies. So we say drug is a substance that alters the normal functioning of the living organisms, and they have side effects. Uh, they have a specific effect and interact with the biological systems. Okay. Uh, so basically, that is what a drug is. Properties of an ideal drug set therapeutic objectives, the maximum benefit within the minimum harm. So one of the properties of an ideal drug is that these drugs will be able to achieve the therapeutic objective. Before we give a drug to someone, we already know that we need this drug to be able to help this particular person in this one way. But then we need this to happen while we are minimizing side effects or harm. So to be able to obtain the therapeutic goal while maximizing the benefit and minimizing the harm. Okay. These drugs will also be effective. That means the drug elicits the desired responses. Uh, there's also be safety. Okay, we need minimal harmful effects, though ideally it is none. But at least the aim is that the drug should be safe. So the safety of the drug means it is able to achieve its therapeutic objective with also minimal harmful effects. Selectivity elicits only the desired response. Okay, so if we are giving an antimalarial to an individual, it should only be able to treat this antimalarial, this malarial parasite that's in the body, and not just to cause other uh, different response that we do not intend in the human body. So it should be selective to what it's intended to do. Reversibility, the action subsides appropriately. So once like the drug has performed its use, okay, um, the action should be over. Okay, the drug should be eliminated and its action should not be long lasting in the body because it is intended for a specific purpose within a specific period of time. Predictability. So we need consistent patient response. So we need to be able to predict what happens in the person once he is being observed on this particular drug. It should be a predictable pattern, not just like we treat a patient with the same drug is a different response, another patient is a different response. That one means we may not be able to understand this particular drug, and it makes us worry, and we may not. It may not be reliable and predictable. Ease of administration. You might realize this is one of the common problems. Uh, Many of us find it easier to take drugs through the oral route because it is easy self-administering and easy to take as compared to the IV medications that first of all may need like professional trained personnel and they are not fit for home treatment because perhaps at home we don't have the facilities and the trained personnel to do so. So that's why preferably it is easy to use the oral medications convenient because even at home people are able to take care of their treatments. Properties of an ideal drug containing there should be freedom from drug or drug interactions. We shall look more at this in the coming slide. It should also be low cost and chemical stability. So we all know the cost associated with drugs, especially those who are on chronic illness and the medication they are taking. We just get to know that medications are getting expensive. Simple generic name for effective communication. So again, this is one of the important points. The same drug should be known, if not worldwide, with just the same name, for ease of uniformity and for ease of people understanding its uses and the diseases that it treats. Note the ideal drug is a theoretical concept. No drugs possesses all of these particular properties. Now, let's look at generic versus trade names. So you might have already been familiar with this, that sometimes for the same drug we find different names, and this happens just because different manufacturers rise to use different brand names to be able to identify the products of one manufacturer from the other. But normally for uniformity, there's what we call the generic names, which is the name that goes across for all the drug universally. We are supposed to know this drug with the same name. Okay. So for example, we have given an example. We have acetaminophen. Acetaminophen, you might be knowing this, the common paracetamol name, which is a bit common. Okay, it's actually the same constituent in the acetaminophen. The trade names may change, but patients may also be familiar with them. But back to some of these trade names, you might have also been familiar with aspirin. The aspirin that we use currently, the name aspirin actually is it was a generic name. I mean it's a trade name. Sorry. Okay. So the generic name for aspirin is acetyl salicylic acid. But then this name aspirin was used initially as a trade name, but it has almost taken over, and we are not almost more familiar with actually the generic name, which is supposed to be the most universal one. So you will have to get to know that for us, the clinicians, we should always be familiar with the generic name because this is the language that we are allowed to communicate with. I see many people prescribing which are trade names, but sometimes it becomes a bit difficult for patients to find these particular drugs in other pharmacies. So we are supposed to use the generic name for easy ability. We look at pharmacology and chemical drug use. So here we look at how drugs produce therapeutic effects and the adverse effects, the unwanted effects. Reasons for the drug selection for a specific person, dosage route and central of administration, decision, strategies for prompting beneficial drug effects, strategies for minimizing undesired effects, and also providing drug-related patient educations. Let's look at pharmacology versus toxicology. What's the difference? So pharmacology encompasses the drug effects and the clinical use. Toxicology focuses on adverse effects of toxins and drugs in excess. So as the name suggests, we are looking at the toxic side. Okay. Actually, we said every drug is a poison. What only makes it not a poison is the dose. When you use the right dosage, and then this is not a poisonous drug. If we use overdose, then it's already a poison. So it's a balance of pharmacologic effects versus the toxicologic effects that uh, no identical response. The individual variations in drug response research, no identical responses, no two patient respond identically to the same drug regimen. So the point here is that even if we give the same drug to different people, we don't expect them to exactly really respond in the same way. This is in terms of sometimes how someone feels side effect of the same drug, and maybe somebody feels mild or somebody even doesn't feel any side effect. That's because our systems adapts differently to the same drug. Factors affecting drug response. One is the drug-drug interactions. Okay. Two drugs interact with each other taken at the same time. This will produce different results. Physiological variables like age, gender, weight. So we all know this very well that the age plays a role in the prescription of drug. Also gender and even the weight. Weight is very important, and that's why we always in our prescription we give drugs according to milligram a kilogram body weight. You might appreciate this more, especially in pediatric, which is really we are a bit more specific because we are to tailor the treatment according to their ages. Pathological variables, we have reduced elimination with liver or kidney dysfunction. So one point here is that we always have caution when we are prescribing to people with already known kidney or liver disease. Why? Because we know that uh, some of these drugs or that are eliminated through the kidney may not be eliminated in a better frequency than in the normal person because already the kidney has a dysfunction, and then we are giving this person this particular drug, so we need to consider the dosage we are giving. Otherwise, we might always be uh, putting them at the risk of injury because the kidney is not doing its function, or the liver is not doing its function, and these drugs remain unremoved in the body, and their toxicologic effects are felt. Genetic factors altering drug metabolism and causing unique reactions is also one of the things we consider. So Taylor in drug therapy said pharmacotherapy must be personalized for each individual's needs. So back to the point, if we are dealing with all these categories, we need to be considerate of how we are going to treat them and the dosages that we are prescribing. Drug development. So in drug development, it's a complex and systematic process involving discovering, designing, and testing the new medications. This one aims to bring safe and effective drugs to the market to address unmet medical needs and improve patient outcomes. So why are drugs developed? Drugs are developed to address various healthcare challenges, including treating diseases, managing symptoms, preventing illnesses like our vaccines, improving patient outcomes, and enhancing the quality of life. The development of new drugs is driven by the need for innovative therapies and also advancement in medical science. Let's look at the preclinical drug discovery. The preclinical drug discovery is the initial phase of drug development, which involves laboratory-based research and testing. This one includes targets identification, a lead compound identification, and initial testing in cell cultures and animal models. So the point here is that in the initial stages of drug development, we always have to use animal models to study this particular drug before it is tried in humans, so that we are able to understand what it does, what are the harmful effects, how does it work. Pharmacological profiling. This one evaluates the effects and mechanisms of action of a drug candidate on biological systems. It helps to determine a drug's therapeutic potential, efficacy, and safety profile before advancing to the clinical trials. So, that means before we start a clinical trial, be it a randomized control trial, uh, the first thing we need to do is we have to do what we call the pharmacological profiling, which is an evaluatory process. Okay, this one evaluates the effects and mechanism of action of the drug candidates on the biological system. How does the drug work in our biological? I mean, in the biological systems. This will help us determine the drug's therapeutic potential, and after that, yeah, efficacy and safety profile, and after that, we can now do our clinical trials. Safety and toxicity testing. Safety and toxicity testing is an essential step in a drug development to assess the drug candidate's potential adverse effects and safety profile. It involves conducting various studies, including acute toxicity, subchronic toxicity, genotoxicity, and reproductive toxicity testing. Clinical drug development, you said this one involves testing the safety, efficacy, and optimal dosing of a drug candidate in human subjects. So here we are getting more concerned about maybe which dosage works. It consists of several phases designed to gather specific information about the drug's effects and assess its overall benefit risk profile. Let's look at the stages of drug development. We have the phase 1, phase 2, phase 3, and phase 4. Phase 1 is specifically about drug safety. It is dedicated to safety testing. How safe is this drug? And then phase 2 is now about the efficacy. Okay, how efficient the drug works. Phase 3, now we go for large scale trials and to be able to see how this works in randomized in a large group of study subjects. Okay, and then the fourth one is now we are doing post market surveillance. Already we are trying to see how uh, we can roll this out at a large scale, market scale, so that the drug uh, is known and people might be able to access it. You know, in making all these drugs, there is what we call assessment of uh, the, the ability for the community to buy the drugs. Okay, so If this is a drug that we are going to manufacture at large scale, then we also need to see that people are aware, and then they'll be able to buy the particular drug for the treatment of conditions that are already known, but maybe the drug is still new in the market, so we need to do this post-market surveillance. Phase one safety testing. In this phase, the drug is tested on healthy volunteers. Okay. So what we do in phase one is they are healthy volunteers. The primary objective is to ensure that the drug safety, the drug is safe for human consumption. So the key point here is that we have to focus on safety assessment in phase one, and then the involvement of healthy volunteers, and then initial step to confirm the drug safety profile. Basically, these are already patients that are already having the disease, and if you get to know, like in large research hospitals that handle research facilities, these particular patients will always be talked to, and with their consent, uh, these drugs can be used because already they are suffering from the disease, but also this drug will give a high hope of improvement, perhaps, and also after that, it will become a basis of measurement of how best the drug will be able to help potential patients in the future. Phase two is the efficacy testing. We said trials here involve human subjects with the targeted disease. The emphasis shifts to evaluating the drug's effectiveness. Key point is persistence of the disease and the drug is intended to treat, and then assessing the drug's efficacy and potential benefits. Phase three is now the large scale trials. So large scale trials conducted in medical research centers aims to gather comprehensive data, especially on infrequent and rare adverse effects. Key points here involves a large number of patients. Normally, we put more patients in this uh, clinical randomized control trials. Provides information on rare adverse effects. So this will be found through the study process of this large number of our patients. And then drug approval is based on satisfactory phase 3 studies. After passing this stage, and the results are satisfactory, that means this drug can already be approved by the Food and Drug Administration Authority. Phase 4 is the post-market surveillance, voluntary phase, focusing on post-market surveillance of the drug's therapeutic effects after phase 3 completion. Key point is post-approval monitoring. It involves feedback from doctors and also health professionals, and then detects long-term therapeutic results and adverse effects. Objectives of clinical drug development. Here, we are interested in assessing the safety and efficacy of the drug in the intended patient population. We also determine the optimal dosage, administration, and treatment duration, identifying and managing adverse effects and drug interactions, and generating data for regulatory approval and market authorization. Ethics in clinical trials. Ethics in clinical trials is very crucial to protect human subject rights, safety, and welfare. So as we get to know the issue of ethics in the professional medical ethics, that some of you have already learned through is really key, and this one cuts across to any study or any research that you will be doing. And if this research encompasses it uses human subject as part of the study, then there are ethical uh, considerations that we will have to undergo through the institutional review board, the research body, governing body. You will have to comply with all these ethics before any study is carried out or before any clinical trial is carried out. Fundamental ethical principles include non-malfeasance. This one means do not harm. Okay, non-malfeasance do not harm to the patients or the participants who are in this randomized control trial. Beneficence is that shall maximize benefits and minimize the risk. There's justice, fair distribution of the benefits and burden of the research, and also dignity. We have to treat these people with dignity, respect for a person's dignity. So obtain informed consent and respect autonomy. Phases of clinical trials. Clinical trials are conducted in different phases to gather specific information about the drug safety, efficacy, and optimal use. These phases include phase one, it's a small scale studies to assess safety and dosage. Phase two, larger studies to evaluate efficacy and side effects. Phase three is also large scale studies to confirm effectiveness and monitors adverse reaction. Phase four is post marketing surveillance to assess long term safety and rare adverse events. Limitations of clinical drug development. We said um, the clinical drug development has certain limitations, including limited sample size sometimes. The clinical trials typically involve a limited number of participants, which may not fully represent the diversity of the patient population. You will get to know about sampling method and also how we approximate our sample to be able to be representative enough for the entire population. And this is exactly what this point is trying to say. If you can reflect back from your biostatistics, or if you are yet to learn on that, you will get more about the importance of an appropriate sample size as a representative portion of the entire population. More limitation, we have time and cost constraints. So conducting clinical trials is time consuming and expensive, which can limit the number of drugs that can be developed and tested. Okay, most of this takes like 5 years plus to develop just a single drug to complete the whole of this process. So you imagine what will have happened. An example I will give here is we did hepatitis E vaccination in South Sudan in Bentiu last year. Uh, this vaccine was manufactured by a company in China called Kekolin, and it's licensed for people who are 16 years and above. But when we were doing the studies, we found that hepatitis E affected even quite a large number of children who are under 16 years. So the point is that, okay, we have vaccine only for adults and we don't have vaccine for under 5 children, I mean under 16 years old, who are also more affected with the disease. So how best can we solve this? But we cannot have an immediate answer to this because if we need this to happen, we need to start to study again the vaccine to develop a vaccine that fits this age group. And we need to start from now and maybe take like 5 years to be able to come up with a solution. So it's not a quick thing that we can easily do always. Ethical consideration, said ethical consideration such as patient recruitment, informed consent, and balancing risks and benefits can pose challenges during clinical trials. We already look at the institutional review board and the ethical considerations. So how to appraise a clinical trial? Appraising a clinical trial involves critical evaluating its design, methodology, results, and implications. The study population, randomization methods, blinding, outcome measures, statistical analysis, and potential biases are key factors to consider. So this in short is trying to give us, in general, the steps that we do in conducting a clinical trial. We have to first select our study population. Okay, who are the people? Is it a community in South Sudan? Okay, and then what will be our study sample then that will be representative of the whole of this population? And then how are we going to randomize? It has to be randomized because randomizing minimizes the chances of bias. So it has to be random allocation of participant of study subjects into groups. If this is a cohort, we have I mean, this is a group that is getting the intervention, and then also the group that is not getting the intervention. But then this will be at random, so that it minimizes the bias. Blinding. So blinding means that participants in the different groups uh, doesn't know what is happening with the other group. Okay, so that it minimizes also the bias. And then the outcome measure statistical analysis. Of course, it involves a lot of statistical analysis to be able to give concrete and reliable results. And then, of course, still the risk of bias is there, but we have to be always cautious so that our results carries the best results to minimize bias as 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 maximum as we can to make sure it is free from bias. Statistical design, we said statistical design is in clinical trial involve planning the sample size, randomizing, blinding, making the analysis methods to ensure reliable and meaningful results. And this helps determine the power of the study and to detect the difference between treatment groups. Parallel group and crossover trials. The parallel group and crossover trials are standard designs used in clinical trials. The parallel group trials involve assigning participants to different treatment arms. Okay, so we have the different groups, and then we assign the treatments. I mean the participants to these particular groups. While crossover trial involve participants sequentially receiving multiple treatments. Randomization and masking. So randomization involves randomly assigning participants to treatment groups, reducing bias. For example, when you select something at random, it means you had no predefined criteria, so it occurs by chance, and then it minimizes bias and ensuring a balanced distribution of patient characteristics. So to minimize bias, masking, blinding refers to keeping participants, researchers, or evaluators unaware of the treatment allocation. Primary outcome in a clinical trial is the principal measure used to assess the treatment's effectiveness. It should be relevant, objective, and reliable, and is often selected based on its clinical importance and ability to reflect the treatment's impact. Okay, so the primary outcome is the most important and the most reliable. Okay, and is based on the clinical importance and reflects on the treatment's impact. Power calculation determines the sample size needed to detect a statistical significant difference between treatment groups. It considers desired significant level, effect size, variability, and the statistical power. So we have come to the end of our presentation. Thank you for being with me. See you in the next lecture. Bye bye.